Production of polio vaccine. Polio vaccine is given for the prevention of poliomyelitis disease that affects the central nervous system of humans. This vaccine is available in two forms, in killed form called as Salk vaccine, whereas in live attenuated form called as Sabin type of vaccine. The names of the vaccines are given according to their inventors. Salk vaccine was invented by Jonas Salk and it is given intramuscularly, whereas Sabin vaccine is a live attenuated vaccine developed by Albert Sabin and it is an oral vaccine. Along with the contributions of these two scientists, there is a Russian virologist named Hilary Kaprowski. He has also contributed in the development of oral polio vaccine. The major difference between Salk and Sabin vaccine is that Sabin vaccine, being a live vaccine, it follows the natural route of infection of virus, that is through intestinal tract, thus results in development of mild fever followed by development of immunity. This vaccine is produced as trivalent vaccine using three different types of virulent strains of polioviruses that represents three antigenic groups or serotypes. This is the picture of Jonas Salk towards left vaccinating a boy named Randy K. A 6-year-old boy is the first recipient of first Salk polio vaccine in 1954. Towards left it is Albert Sabin and towards right it is Hilary Kaprowski. These are the pioneering virologists who developed oral polio vaccine which we are using nowadays. Production of Salk vaccine, that is production of killed polio vaccine. The first step is the growth of viruses, that is cultivation of viruses in the medium. For this, 5 to 10 liters of steam sterilized closed bottles as shown in the figure or glass jars or roller bottles containing sterile nutrient medium called as 199 medium 199. It is for the growth of animal cells. For this, monkey kidney tissues are used as a source of tissue culture. Then inoculation with monkey kidney tissues. The tissues are mixed in sterile glass tubes and stored in frozen conditions in liquid nitrogen, that is at temperature below minus 100 degrees Celsius until its use. These monkey kidney tissues are then inoculated into the above mentioned medium and incubated on the rocker in a temperature controlled room for 6 days for the growth of tissues. The rocker provides continuous mixing like this. After incubation, the bottles are added with fresh nutrient medium and inoculated with 3 serotypes of polioviruses separately. For the large scale production, this fermentation is carried out in a bioreactor of approximately capacity of 1000 liters to 3300 liters. This bioreactor is operated with automatic control system as this involves animal cells that are highly sensitive to shearing stresses. This is an example. This fermentation is an example of animal cell fermentation and the typical feature of this fermentation is its sensitivity towards shearing conditions. The simple reason behind that is animal cell doesn't have cell wall and that's why they are highly sensitive to shearing conditions. In the next step, the contents are incubated for next 4 days during which the virus multiplies 250 to 100 times, 1000 times. The multiplication of viruses is checked by observing the cytopathic effects on the cell lines, which is done microscopically. After the completion of life cycle, the viruses disrupt the cells, releasing the viruses into the medium, which are then harvested. Harvesting Harvesting a viral suspension is done using two steps like centrifugation and filtration. Centrifugation and filtration will remove the animal cell debris and contamination. Filtration is done through bacteriological filters for the separation of bacterial contaminants. The concentration. Next step is concentration. This viral suspension is then 500 times concentrated by the method of ultrafiltration. Ultrafiltration is a method of separation of biomolecules on the basis of its molecular size. That's why it is highly selective method for concentration. After concentration, next step is purification. Purification of this viral suspension is carried out by using gel filtration technique on a Zephyrus column or with the use of ion exchange chromatography. During all these operations, there is a regular checking of viral titer. That means the amount of viruses, the concentration of viruses in the viral suspension. So this is carried out during the whole process, and the methods employed are ELISA or radioimmunoassay, and this assay is done using standard antibodies against the viral antigen. Next step is inactivation. The concentrated purified viral suspension is then inactivated by a chemical called as formalin at a concentration of 1:to 4,000. Incubation with constant stirring helps the process of inactivation, and this continues for 66 hours to 12 days at a constant temperature of 37 degrees Celsius. Then there is periodic checking for the presence of live viruses. Obviously, this treatment is going to kill the viruses. The next step is blending. In simple words, blending means mixing of different viral components. That is, mixing of three viral types which were prepared separately. Now they are mixed to form a bulk in a large vessel. A neutralizing agent such as sodium metabisulfite is added along with thiomersal for the sake of preservation, long-term preservation. Fifth step is filling and drying. Filling is done in sterile ampoules or vials and freeze dried, followed by sealing. Sixth step is potency test. The preparation is tested for its potency, means ability. Potency means its potential by inoculation in chicks or guinea pigs, followed by measuring the antibody titer. Last step is safety test. It is performed by inoculating the cell culture and monkey spinal cord with this final preparation, and then the animals are kept under observation for the development of any clinical symptoms for several days. This is the flow sheet for